being into that 3 years, I think you were always every day you were struggling to get a success. How those day looks like?
Every day we're trying different conditions, different
But in that process, you never had got a thought of give it up और maybe चल छोड़ देते हैं यार इसको.
Industry doesn't work the way academician work. Your every result has to be validated thoroughly.
That time also bioprocess experts were very niche, right? In between you talked about biosimilars. So I think biosimilars is nowadays lot of buzz in India. So let's talk about what's actual reality and what actual buzz. So how India is doing in biosimilars?
India is doing really great in terms of biosimilars. But if you look at the market, India has the highest number of biosimilars already approved. I think around 98.
98, yeah. So what's your one message to the young generation who wants to get into biopharma, biologics? So what would be your one piece of advice? Hello, Dr. Patra. Welcome to the Pharma Now. So first of all, thanks a lot for coming here and giving your time for us. I think your journey, I was just trying to go through your journey. It's mammoth. So it's, I think, more than a couple of decades of experience now, right? And I think you are one who has been a very initial person or initial leader who was setting up the bioprocesses in India. So let's start with the start.
Okay.
Yeah. So I wanted to really understand. So where are you coming from? Like from Orissa or what area?
Basically, born and brought up in Orissa.
Okay.
So in my education till masters, it's from Orissa. So in fact, before this, when we are discussing, you're asking when, what exactly the initiation point of getting into biologics.
Yeah.
So I was trying to recollect what happened, why I get into biology. So there was a time when I was doing graduation in one of the famous college in BJP college in Bhubaneswar.
Okay.
So my teacher, Dr. Shiva Prasad Mohanty, that time he was there, he was a very good teacher. He used to teach very nicely. So he used to take care of our zoology. We are into zoology honors.
Okay.
That since then, he was telling us how to do business utilizing a biological resources.
Okay.
That time we were back in 1986. He was telling how to do vermicomposting in a large scale. So that's the initial stage of bioprocessing, the vermicomposting, using worms, developing a compost, etc. So that was the initiation point when I was, it was really getting fascination. I was really getting fascinated towards this, how to utilize the bugs, the worms, and produce different things. And of course, after that, I started my MSc in life sciences, studied genetics and molecular biology as my specialization, and understood how bacteria actually plays a key role in our whole internal system, particularly anatomy and all. After that, in between, I had a couple of years of experience as a lecturer, and then I went for PhD. That's in Northeastern Hill University in Shillong.
Okay.
And then from there, I went to NII, that's National Institute of Immunology, where I started my first postdoc.
Okay.
The first actual bioprocess started there.
Okay.
That time, Dr. Panda was there. Dr. Amulya Panda, he was a scientist in NII, National Institute of Immunology. He retired as a director of National Institute of Immunology. So the lab I used to call as a product development lab. And if you know Dr. Talwar's one of the vision is to bring a vaccine against anti-fertility vaccine, basically, against HCG, human chorionic gonadotropin. And the whole institute was built to make a vaccine. Of course, a lot of research had been done that time, but the process setting was difficult. So apart from that, then we were doing other processes, one of them was human growth hormone, ovine growth hormone, bovine growth hormones, all few different growth hormones at that time we were doing. And that was the initiation time when we were using one of the primitive bio a fermenter. Um, and we started processing, we started from 2-liter scales. Initially, it was a really challenge to produce huge amount of growth hormones that way back in 98 time. So, and I learned how the bioprocessing has to be done. Controlling parameters like oxygen, pH, pressure, feed strategy, everything I learned from there. Dr. Panda, he was the one of the best teacher, I would say, and he taught me how to do the process optimization, etc. And in that lab, that time, getting around people used to get around 10, 20 ODs in microbial culture. So we could set up a process with as high as 50 OD that time.
Oh,
Of course, 50 OD at this moment in microbial culture is not great now. But that time it used to be very high.
correct. In 1990s.
In 1998, I'm talking about
Yeah.
1998. So, and not many microbial laboratories were there with such a high OD.
Right.
And further processing and downstream processing was also difficult. People used to produce in insoluble format, but we used to produce in inclusion bodies. Because if you start producing in a very high rate, protein tend to get into inclusion bodies. So the next challenge was how to purify the proteins from the inclusion bodies. So we produce a lot of protein, but how to purify them? They are all aggregates. So we have to develop a process. Then downstream processing started. So initially the upstream process, take the challenge, take the OD, increase the OD, produce, but then the downstream challenges came in. And getting inclusion bodies at that high rate was kind of it's a huge amount of protein in inclusion body. So we started developing process in downstream processes. Right? A lot of effort has been put in, and we're fortunate enough to get a publication out of that time, and we could get nearly about 25% of the protein inclusion bodies, which at that time, during I think more than that, 25 or around 40% of the protein, because the in culture you'll only see inclusion bodies there, so the protein, cellular protein will not be there after initial. So, and the process was optimized in such a way from inclusion body you produce the protein, and protein was pure, soluble, active, and volume of the productivity also pretty high at the rate of around 30% if I remember correctly. And we published a paper in refolding and characterization of human growth hormone, and that particular publication, it's I think so far has been cited more than 300 times.
Oh,
So that is one of the biggest achievement, and of course followed up a lot of other works has been done. Bovine growth hormone done, ovine growth hormone done, but everybody follows the similar kind of procedure. Initial process, maybe a little bit of modification,
correct.
some other components, yes, and same process. Now, if I remember correctly, now we go up to 40 OD just like that. With little bit of optimization, the cell density goes up to nearly 100 plus 150 OD. So if you look at the fermenter vessel, you look like the Rava Suji. It's like that. so much of cells,
Okay.
then
But this was your academic life. We
basically,
switched to industry.
and as at that time in 90s, everybody has a trained for US or any other countries for
Right.
that.
Right.
NI was kind of a lunch pad for us. 2 years of postdoc and then go abroad.
Hmm.
So,
But that was a template. That is a template. I still it is
getting followed. A lot of people they use that one. Then I went to US, worked in because of in this credibility, producing a lot of protein in inclusion bodies and purifying so much of protein. So one of the professors selected me and then I went. But little did I know that it's not a kick up, when a bed approaches that you lie down.
correct.
It's not the path is not that easy.
Yeah.
I may be very successful, getting human growth hormone, at that high rate, or bovine growth hormone at that high rate. So when I started working in Worcester Polytechnic Institute, that's in Massachusetts, the problem was it was a different protein. The source was different.
Okay.
The source was archaea. Archaea, the bacteria which are growing in hot springs.
Okay.
The temperature is around 75, 80 degree.
Okay.
There are some bacteria that grow in those conditions. And the protein, what I worked is copper transportase. So even though we recommend until try to express, but the conditions were different. It's supposed to grow at 75 degree, but we are trying to express them in 37 degree. Didn't grow. So that was a big setback. We tried several different ways, methods, protein didn't grow. And the professor got fed up. Forget Ashok. I don't think it's going to happen. So I said, okay, fine, no problem. So I switched to another university. That's Boston College. So they are things were initially okay, fine. The protein was expressing fantastically, and again the bioprocess was good. Everything remained same. Protein expressed, protein soluble, protein purified. That is another challenge. Getting an active protein. So getting active protein was really a challenge because you may produce it high level, no problem.
Correct.
You can purify it, but getting active protein is a challenge. You have to process, optimize in such a way so that at the end, our protein, what you are getting is actually active. Because otherwise, if the protein is not active, non-functional, no use, whether it is a normal research work or even for therapeutics.
Correct.
The protein at that time I was working is actually as a cancer target. It's a aspartate carbamylase. So that protein it has lot of challenges. So we try to address those things. Couple of years struggled, and protein the activity was restored, but not to the expected levels. Somehow we managed to form protein. I mean it was an enzyme it's a it's supposed to have a similar aggregation of both regulatory and catalytic domains. They should come together and form the whole protein. We could do it that, but then the level of activity went down. And step by step I was learning through my educational career. First one, learn how to do the fermentation. Initially they produce the protein fine, he produced. Second, purify. Challenge was that the purification. Somehow I managed to purify. Second, I'm getting an active protein. But then when I came to, after that 2 years, my 3-year visa was over. And of course, I had done another offer from Roseville Cancer Institute in Chicago, in Buffalo. So then he told me to join. Then I said, it's 3 years. I'm out from India. Then I said, okay, let me go back. I came and joined Ranbaxy that time.
Okay. So being into that 3 years, I think you were always every day you were struggling to get a success. How those day looks like?
Yeah, I mean, basically, like, that was, those days were a young ages.
Yeah.
And being a researcher, we used to work, when I was in Shillong doing my PhD, we used to work sometimes overnight without sleep. And continuously, like, if there is a problem, like, let's, at that time, like, no automation. So 1992, I'm talking about 92, 93, no automation, nothing. So everything you have to do manually. So that's why, like, sometimes if the work is going on, if I have set up my experiments and I have to complete those experiments, I cannot leave it in between. So I have to stay overnight.
Yeah, overnight. Yeah.
So 18, 20 hours in 24 hours throughout, like, till I completed my postdocs, both US as well as in India. So it was kind of like, I wouldn't say struggling in terms of finance, but struggling in terms of, like, getting the desired result. When you have, like, fixed target, okay. For example, like, I had a fixed target to get that particular, like, aspartate tablet, like, in highly active protein. I could achieve maybe around, like, 50%, not 75%, not 100%. So not even 90%, so that I could recycle. So this is, though in that way, yes, it was struggling. Every day we're trying different conditions, different
But in that process, you never had got a thought of give it up or maybe चल छोड़ देते हैं यार इसको.
No, that didn't come from my side. It came from my boss side. So at one point of time, he said, "Ki Ashok, like, leave it." Like in first stage, when I was working on copper transporter, he said, Ashok, leave it. Like, you have been trying so hard, nothing is happening. So there's a problem. Like, the problem we identified is the archaea. What we are doing is, like, it's supposed to grow at 75 degree, and we're trying to grow that in 37 degree. So that is a challenge. And then some modification has to be done. So, and then in second postdoc also in the US, so same thing happened. Like, even though I could finish the work, I could, with a little bit of less activity, some of the results I could get. It's okay. But then our interest was, like, to target those enzymes as an antigens. That didn't happen. Okay. So that is, yes, that is a failure. We didn't get it, but research always has a failure. It's not that every day you get a result which can take you to, like, a product. So that's how I learned, like, it's, you have to build upon your failures. As I said, like, starting from the beginning, like, I started learning things step by step.
Correct.
More I went towards, like, complete process development things. Initially, it was looking, oh, it was easy, like, it can be done. What is in there? Then every day there is a problem. Every day there is a problem. You change buffer, there's a problem.
Correct.
Composition of the buffer, there's a problem. So all these things, that time, all those days, like, it was a kind of what we call the OFAT, one factor at a time. Now the same thing people are doing, it's like, with a machine learning. Data is available, or you design a data, fit into the machine is going to learn what is the best condition.
Correct.
And then give you a solution of 5 different or 6 different choices, which can easily be managed. Correct. Rather than doing, like, millions of very
Basically simulate the process and it gives you the probable options.
Exactly.
Right. So though you came back to Ranbaxy.
Ranbaxy, and that time, like, they were focusing on, like, small molecules. In fact, Ranbaxy was always a small molecule manufacturing company. But R&D was there. R&D was great. It was the able leadership of, like, Dr. Rasmi Barbaya and Dr. Kasim. They were, like, good, very good people. And that time we were working on pharma division has a responsibility to produce one molecule. And the molecules what I was working on, basically I am not a chemist, so I was a kind of supporting staff to the molecule development. There, my role was to develop kind of cell lines. With those cell lines can be utilized for the assay of these molecules. So it's industry. That time I came to know the industry research is totally different than academic research.
Right.
In academics, you have a single project, you take it forward and go. Whereas in industry, you work on a single factor of a particular project. Because from the academic point of view, when you look at developing a particular molecule, then you just do initial research, study in vitro efficacy, in vitro potency in all cellular conditions, small volume, and say, okay, my molecule is working. It's a very potential drug. I'll patent it and then I'll sell it. But when it comes to industry works in a different mode altogether. There I came to know the first learning is that yes, industry doesn't work the way academician work. Your every result has to be validated thoroughly in multiple conditions. It's not just one condition. In academic, we just see one condition and it's fine. But in industry, it has to be tested in multiple conditions. It has to be validated in several different things, and then cross validation has to be done. Multiple lab validation has to be done. Then only say, okay, this is working.
Correct.
Then in industry, I was taking care of cell line developments. That was my expertise and my strength. So cell line, I started developing cell lines which were used for assaying the small molecules at a small scale. So that was not much of process development at that time. And then after 2 years, of course, we are pretty successful in developing 10 different cell lines for the molecules what I was talking anti-BPH molecule and anti-muscarinic molecules. And then I shifted to Jubilant Biosys. Company changed, different company. I thought, okay, I'll be giving a lot of responsibility.
Basically, NBXC, you were not in bioprocesses.
No. Actually, the real bioprocesses, I after my postdoc in NIA, there's a big gap of actually real bioprocesses till I came back to Panacea.
Okay.
So it's Ranmachi 2 years, Jubilant 2 years. These are all different molecules and different bioprocess. But these things gave me a lot of different kind of experience.
And different perspective.
Different business perspective.
Yeah.
So when I came back to Panacea, it was my field cell line development and bioprocess development. They are the real challenges.
So that time also bioprocess experts were very niche, right?
I won't say niche. It was in 2008. Quite a lot of people were there, pharma people were there. But the process and the way right now the process has developed, that kind of advancement was not there.
Okay.
Even many people didn't know how to do a fed batch processing. So in Panacea, initially the first molecule what we are trying to do is erythropoietin and darbepoietin. So that time the person who was in charge, they were doing only batch processing. So in batch processing, of course, your longevity is not there. You cannot keep a batch more than 10 days. Cells are not going to grow for 10 days in a 10 maximum. So, but the same thing when you change little bit of fed batch process, your cell density goes up, production goes up, and the batch duration also goes up. So starting from 7 days, it can go up to 15 days. So that's a big advantage. So there When in 2008, when I joined, the initial target was this, but in 2009, H1N1 came.
Okay.
When Asia Biotech, being a vaccine company, immediately switched the direction. We want H1N1 vaccine. That was the direction from the chairman. So we started. So what happened, like, in I think, if I remember correctly, 2009 March. Yeah. Okay, we want H1N1 vaccine. And we don't have time. No, we have to do it. So I said, okay, let's try it. With me, Dr. Neeraj Agrawal was there, and he was a virologist. He's working right now in ICMR.
Okay.
He's a virologist, so he said, like, we'll do the process, and in process modification, we'll do it in egg. So, okay, let's do it. The biggest challenge was to develop the downstream processing. Scaling up was not known because no bioreactor, nothing. Eggs with multiple eggs, like, you buy eggs, inject it, virus grows, isolate, purify, and keep it ready, formulate, and your vaccine is ready. It's an attenuated virus, no problem. We set up the process of 5,000 eggs. Initially with 10, 12 eggs started, and R&D completed with 5,000 eggs with a very good titer, single step processing, and it's a column chromatography processing. Process was ready, process transferred to the facility was in Lalru, Punjab, near Chandigarh. So there we scaled it up to 65,000 eggs.
Okay.
The real challenge is how to inoculate them. 5,000 eggs, still we could do it manually. Facility, BSL-3 facility, because you are handling H1N1 vaccines, even though they are attenuated, but you have to take care of that.
Correct.
So when we scaled up, but manual inoculation is not possible, automated inoculation happens. So when automated inoculation happens, automatically, like, suction of the allantoic fluid happens. You cannot, like, manually take out your allantoic fluid from the egg, 65,000 egg in couple of hours. So the real challenge was the yield went down. Started thinking, like, what happened, like, the same process, so why the yield went down? So when we started asking, like, what is the exact reason, could find out that when we do it from manually, we could take out around 8 to 10 ml of allantoic fluid from the egg, but in automatic syringe, it could pull out only 6 ml.
Oh.
It doesn't go deep.
Right. Yeah.
And if you put it down, then it may sub the egg yellow.
Yeah.
And so that is a big challenge, how to do it, how to overcome that one. So I said, we have to take care of that one, because 6 ml fluid you are throwing out. So that is almost 4 ml fluid you are throwing out, a 40% loss. And then somehow, like, some adjustment were done so that it can went further in. But we had to compromise with that, yes. Beyond that, we cannot go. If we go, then we have a lot of impurities, and then from 65,000 eggs, column purification, so we could produce the egg. The process was good. The process was like, we filed a patent for the process, and then Vanashya was happy, kind of, okay, we have a process. But by that time, it all happened in 6 to 7 months time. Really working hard that time, like, right night at 1 or 2 o'clock, come back to home, and again in the morning by 9 o'clock you were in the office. go to production lab, demonstrate, address the problems, find out the solutions, why this that happened. Some then by the time it was, I think October, November, so process was ready, and then commercialization process set up. Then there are some other issues came up, how we are going to commercialize this, and there is some problem, this problem, that problem, not benefit, not making any money. I said, see, as an R&D scientist, whatever I could have done, I have done it. And how you're going to commercialize it, you say it. So, no, you with this much of title will not be able to do that. Let us find, what else I can do. Otherwise, it could have been a good product in the market at that time. Then, that time the chairman took a decision not to put it in the market. Fine. So, and we kept on working on some other development, biosimilar development. And then in the meantime, I met Dr. Singh. So he said, Ashok, I have a vision that we should, he himself is a very good scientist. So he said, I want to set up a small facility where we can develop biosimilars or biologics, either novel biologics or biosimilars. I said, okay, it's my hometown. I'd be happy to come and help you set up all this facility. Then came back here, started working, both bench as well as other things. Since then, I have been working, and trying to get some funds from the government and set up a small R&D lab to develop the processes. Everything is done at R&D scale, not at the pilot scale or even DMP. So it's all 2 to 5 liter scales process being developed. We worked on developing the first molecule what we developed is trastuzumab. But again, it was my bad luck. Everything was done, the process, the RCGM, and we're about to go to preclinical tox study. COVID came in.
Oh, okay.
Yeah, so we produced the material for PCT, and because of COVID, we couldn't do anything, and everything was shut down. 6-7 months or more than that, and our product was kind of finally you had to throw it out.
Okay.
Well, then of course, that's a big investment. That was time. So since then we changed. Let's don't take molecules to all the way to PCT. We'll initially develop the molecules, show the potency and efficacy, complete physicochemical characterizations, and then initial process development and R&D skills, and look for partners.
Okay.
So that is what is the strategy now of IMGENEX. So we are currently working on 2 novel molecules, and one is a IL-17 antibody. That is a novel molecule, but it's a kind of me too molecule, because already the similar antibodies are already existing in the market. But it's a novel molecule in the sense it came out from our own lab. Because we are antibody producing company for reagent. From there itself, we try to find out from the molecules. We are still doing similar kind of research. Whatever monoclonal antibody we make, we try to identify if any potential molecule is there, which can be converted to a therapeutic one. Another one, what we are working on on anti hyperlipidemia molecule. That is basically a modified molecule of the existing antibody. That's one antibody. It's called ivolocumab. That is hypercholesterol, targeting to hypercholesteremia. So it's an antibody. And then what I have done, we did some research in silico activity search, and then we designed a small fragment of this antibody. So that currently is going on. These 2 molecules are currently going on. Apart from that, we have a plan of working on a couple of novel molecules.
Okay.
One is a KL1, another one is a KLH1. So those are at a very initial, just kind of design stage.
Correct. So in between, you talked about biosimilars. So I think biosimilars is nowadays lot of buzz in India. So let's talk about what's actual reality and what actual buzz. So how India is doing in biosimilars?
India is doing really great in terms of biosimilars.
Hmm.
But if you look at the market, India has the highest number of biosimilars already approved. I think around 98.
98, yeah.
So that it clearly indicates India has a big market for it. And a lot of companies are working on that. You name most of the company big pharma companies, Dr. Reddy's, Biocons, Ipca, or Cipla or Zydus. They are doing pretty well. A lot of biosimilars. Intas has a lot of biosimilar. So because you don't need a lot of research. It's the molecule is already existing. The pathway is already established. So you don't have to put a lot of money to identify the new molecules, new pathway, nothing. It's a information is already available. It's a basically same kind of approach the way small molecules
Right.
people are doing. So once they don't know the pharmacophore, do the reverse engineering, change the pharmacophore, or newer modification in pharmacophore, and then produce a molecule. Same thing in biosimilar, you the sequence is known, sequence is off patent, pathway is already established, target is already established. So you have to produce the similarity. But big challenges to counter those kind of challenges, companies Vicon, Dr. Dairies, or even Cipla, Ipca, etc. They are investing a lot of money in in infrastructure upgradation. High-end machineries are coming up, particularly high throughput screenings and analytical instruments employing EPLC, Raman spectroscopy, MAS, SEC MALS, and then your infrared, near infrared, all these things, SPR for binding studies, and machines like Biacore, etc. People are investing all this money, and a lot of money is on these machine.
So is the initial investment or capital investment cost is much higher is a challenge in biosimilars?
Yes, for a small company, yes, it is a challenge. And in fact, the government of India is also facilitating this. What government of India is doing is trying to establish a facility which can provide infrastructures, high-end infrastructure. Like for example, in Bangalore, they have CCAM. CCAM has pretty good facility. NCCS Pune has very good facility. So machines are there, trained professionals are there. So anybody who has some amount of money wants to develop a biosimilar, initial R&D is done, want to do proper characterization, they can approach utilize the facility with us less without investing on infrastructure.
So kind of incubators.
Incubators, you just go and take the service, get your result, and these facilities they provide you GLP level facilities. So there is no question asked, most of the facilities are accredited labs.
Correct.
So whatever data they generate, they are all acceptable from the regulatory and quality control point of quality assurance point of view. So these data are acceptable, and with using those data, you can go to any agencies for approval.
Okay.
So that way, government of India is facilitating all these things.
So what kind of opportunities Indian market or maybe developing countries are offering for biosimilars for the Indian manufacturers?
At this moment, it is bit expensive. All the biosimilars, even though for example, trastuzumab. Initially, it was 1.5 to 2 lakhs, but now 7,000 or 8,000 rupees per dose. So even 7,000 rupees per dose is also not acceptable to many people, for anybody who has a breast cancer. So that is also it's a bit expensive.
Yeah, absolutely.
So but with time it may still come down. In that day, it's becoming slowly very affordable. I won't say like damn cheap, like like capsules or tablets, but still they are affordable. Like because your dose regimen is like for breast cancer is like you take 2 injections per month or maybe 4 injections per month, so 2 to 3 years. So very most of the people who has like insurance, which insurance covers, they can easily afford it.
Correct.
There is a big scope for biosimilars in India, slowly building up.
Sure. So you are also talking about the new technology which are coming in this industry with the AI infusion. A lot of technologies are upgrading. Rather in characterization area also on chromatography also technologies are upgrading very fast.
Very fast.
So what is your take on this digital transformation in a bioprocessing?
It's basically like eliminate the human error. So that's a big advantage.
Yeah.
You cannot keep a person to attend the machine like throughout the day or throughout the night. So once you have digital technologies with AI kind of things, and the errors which are supposed to occur because of human interference is completely eliminated. Sorry, I would say not completely because there could be some malfunction, but to a great extent, it will be eliminated. So that's a big advantage, and of course, ease of operation. So you don't need to put your brain too much. Your process already feed into the machine.
Correct.
And just fill it up and say yes, go.
Correct.
It's all done.
So are you implementing anything, any such kind of technologies at your lab?
Yeah, we are like at the very early stage of development. Yes, we will be planning, but the technology what we are planning to implement is that like high throughput screening kind of things, like the way Humber is doing, like Sartorius Humber machine. So what happened, like initially in manual conditions we could screen maybe around 10, 20 or 50 or different variations. But the new technology, like it can help you operate like all these machines. It is variation, like a lot of variations with hundreds of different variation you can do simultaneously and give a very good data, because all these machines are like comes with inbuilt probes.
Yeah.
So that is a big advantage.
Correct.
But when you do manually, like all these probes are not available, so there is a process related defect would be there.
Correct.
But new machines are coming up, like
Yeah. And I think rather with the extensions of AI simulators, this machine can produce amazing results and can save a hell lot of time.
yes,
So you get create clear simulations. Okay, what models can be generated, and it gives you a few probable combinations, and your researcher can only work on those probable combinations and take it further.
exactly.
So that is really changing the world the way things works, right?
That's right. Yes.
That's wonderful. So I think it's all about science now. I also wanted to understand more towards your leadership mantra, because building this teams, because it's all about teamwork here,
Yeah.
right? And you need to have a motivated people with the patience, with the persistence approach, right? How you keep your team motivated to be on one target for years and years?
You have to find people like that.
Yeah.
With scientific mind bend and uh, with clear cut, like focus on developing a particular thing. So it's difficult to find people like that, and nowadays many people, they come for money. If they don't get enough money, they easily switch. Everywhere, like they get little more and then go. But scientist, the real scientist, like who has a passion to do something, they generally stick to one thing and want to do. And what generally I do when I try to interview people and try to tell them what exactly they should be doing, try to understand what exactly they want. And from the beginning, I try to make them understand, okay, what exactly you are doing. Try to make them understand the process is not like, you put the protein here and then collect it from and
Instant
the column.
application is not possible.
Yeah, it's not like that. When I try to explain them, the difficulties and the variables, people try to understand. So when they understand the process, understand the problems, then they appreciate. And then this very small things, it create interest in them. If you don't know, if you don't know the taste of the coffee, then yeah, once I'll check it and then say, "Yeah, okay, it's not good for me." But if I tell you, what are the ingredients in there and what are the benefit in there, how these chemicals actually work in your body,
Yeah.
then probably you'll be interested to know more about the coffee, different types of coffee. Similarly, when I talk to people, the students which they come to me to understand particularly the processes and both upstream and downstream processes, try to explain them each and every part of it. And I also tell them if you don't do this, what's going to happen. So it's a basically one-to-one discussion that to create more interest in them about that particular process. But that's how people get interested. Very recently, one person who's currently working in our lab, he's into downstream processing. So uh, when people say, okay, now what's the big deal in downstream processing? You have a column, put the solution of the protein and then collect the protein. Uh, I said, okay, fine. I'll give you a sample and do it. Uh, Name the column. I'll give you the column. I said, fine. And he did it that. The result came out not to the expectation. I said, "Could tell me what exactly, what are the variables you think has impacted your result?" Couldn't explain. So I started explaining them. See the variables here. You have a resin. You have to become the right kind of resin. You have to pack the column properly. You have to identify the buffers which are good for your conditions. You have to identify the flow rate, which flow rate is good for your process. You have to identify the pressure also. So all when I started talking to him, then he understood, right?
Step process which you
See,
to approach.
and I told them if you don't do this, if you don't optimize these things, your result will always there. So you think, it's not that you put a sample on top of the column and purify it. You have to design it properly.
Correct.
So that's why the design of experiment is essential. You need to understand. So when you start telling people, the students particularly youngsters, the integrity of these processes, they'll develop interest. But when you don't tell them anything, you'll say कोई ये भाई मेरे लिए processing कर दे ये ऐसा है. Obviously उनका कोई interest ही नहीं है.
Right. Right. So I think in these many years you must have trained, mentored hundreds of researchers, right?
Yeah,
So
okay, it not hundreds, quite a good number of people.
yeah. So is there any story where this mentee has surprised you or make you proud?
Yeah, there are quite a lot of things happens, and yeah, actually I started mentoring people since my postdoc days. Uh, that time people used to come for training. The best thing what happens in one of the student he was very much interested in downstream processing and column packages. So in one of the column which we used to work on is that time it was a I think cation exchange column, I think, if I remember correctly. And, cation exchange resin from the resin was from currently Saitiba, but that time it was Amarsam. So proper packing of the column was very much essential to get the right kind of separations. The, that fellow, I told him, here is a process. You have to purify these two very closely related proteins. So you have to use the cation exchange, but do it in such a way, go step by step, so that we can see what are the problems and why it's not getting resolved, all those things. So and that fellow actually took extra interest, studied the complete physiology and characterization of those beads, the buffers, studied the guidelines of Amazon pharmacy, etc., and then develop a particular buffer utilizing different ingredients, and then did that, and he could nicely separated those two things. So yes, this fellow has a good good researcher mind, and I think he is currently in the US. Yeah.
So this story always gives you a goosebump, like a
And like all of
satisfaction.
people here in I met Jagdish today. Jagdish was also working under me. He is currently in Hyderabad in some yes. He was one of the person who was working under me in Panacea Biotech. Then it's Arun actually. Another person, I didn't meet anybody today except Jagdish. So
That's it. Very interesting. So, what's your one message to the young generation who wants to get into biopharma, biologics? So, what would be your one piece of advice?
keep developing the skills, because with AI and machine learning coming up, the students has to keep up the pace. Apart from hands-on training, they have to learn all these things, and consistency and perseverance is the key. It's failure is going to come definitely. As I told in my life, a lot of failures happened, but it's not a piece of cake. Like you have to solve the problem. Biopharmaceuticals or any processes for that process is always full of surprises. You never know at any point of time you'll bump up and surprise. So don't get disheartened. Correct. Keep working. Failure is a pillar, pillar of success. That's what people say. So
Yeah, that's the very important line. Failure is a pillar of success.
that's right.
So I think Dr. Patra, amazing talking to you. Thanks
Thank
for
you very much.
sharing such amazing insights. I think every story, rather I say ki in science there is no failure. There is always a learning.
Yes.
So all those learning stories are very much important, and I think those gives us more encouragement to move to the new innovation and new milestones. Right. So thanks for sharing this, and I'm sure people will be like this conversation and they will get inspired to get into such kind of innovative and novel area.
Thank you very much.
Perfect. Thanks a lot.